I'm Effie Parks. Welcome to Once Upon a Gene, the podcast. This is a place I created for us to connect and share the stories of our not-so-typical lives. Raising kids who are born with rare genetic syndromes and other types of disabilities can feel pretty isolating. What I know for sure is that when we can hear the triumphs and challenges from others who get it, we can find a lot more laughter, a lot more hope, and feel a lot less alone. I believe there are some magical healing powers that can happen for all of us through sharing our stories, and I'll take all the help I can get.
Once Upon a Gene is proud to be part of Bloodstream Media. Living in a family affected by rare and chronic illness can be isolating, and sometimes the best medicine is connecting to the voices of people who share your experience. This is why Bloodstream Media produces podcasts, blogs, and other forms of content for patients, families, and clinicians impacted by rare and chronic diseases. Visit bloodstreammedia.com to learn more.
Hi there, and welcome to the show. This is Once Upon a Gene, and I'm your host, Effie Parks, and back with another episode that might just blow your brain, organoids. Okay, I don't know how to get that joke in there, but I'm talking with a lovely, lovely lady named Maya from the Brainstorm Therapeutics team, and we're talking about taking a tiny piece of your child's cells and growing a mini version of their brain in a dish. It's very cool. And they're like little clusters of real human brain cells grown from their stem cells. You can study the disease, screen drugs, and really expand on everything that we think is possible for ultra-rare diseases. My guest is young and brilliant and kind, and she's deeply connected to the mission, and she's helping rare orgs move in a strategic way from watching and waiting to perhaps testing and acting. And we talk about how organoids could replace mouse models, how they're helping accelerate drugs toward clinical trials without animal data, and how they might be the key to personalized medicine for our kids and their specific mutations. I hope you enjoy this conversation. I very much did, and it made me think a lot. Please enjoy my conversation with Maya Gosztyla. Hey, Maya, welcome to the podcast.
Hi, happy to be here.
Yes, I'm looking forward to diving deep into brain organoids today. First, can I get a little introduction from you and Brainstorm Therapeutics?
Sure. So my name is Maya Gosztyla, and I'm one of the co-founders of Brainstorm Therapeutics, and my titles here are COO and also the lead scientist. And for my own background, I grew up in Ohio, studied genetics and neuroscience for my bachelor's degree. Have always been really interested in the brain and kind of how it works and what happens when we get a brain disease. And so after I finished my bachelor's, I moved to the NIH in Maryland for a year, worked on how to develop high throughput drug research assays for rare brain disorders. I really loved doing that work, and so decided to go to the opposite end of the country to UC San Diego to get my PhD. And after finishing that work, I had been working on brain organoids specifically, which we can talk more about later, what exactly those are. But that was kind of my main research focus, and so I had the opportunity after I finished my PhD to join the Brainstorm team here. And of course took it. It was exactly the kind of work that I kind of had always wanted to do. So it's been an amazing opportunity to work here and get to have this opportunity shortly after grad school. And in terms of what Brainstorm does, we're a precision neuroscience company. And essentially what we do is we have a platform we've developed that takes human patient stem cells and we differentiate those into brain organoids, which are essentially, you can think of them like a mini brain. They're kind of like a little sphere. They're about 1 millimeter in diameter, so quite small. And they have all of the same types of cells you would see in your brain if you were to take just kind of a little piece of it. And so we can use these as a system to discover new drugs in a model that's as close as you can get pretty much to experimenting on the actual human brain. And so we think this technology, combined with some of the work we're doing with AI to discover new drug targets, is going to allow us to find therapeutics that will actually work when we take them into the clinic.
Do you get offended when people tell you how young and beautiful and smart and accomplished you are? And how is that even possible?
It's been amazing, honestly, to have this opportunity so early in my career. I think a lot of people have to, kind of go through the grind a bit to get to where they want to be. But it was kind of just a right place, right time kind of situation. They were looking for someone with brain organoid experience at Brainstorm, and I was looking to do work with brain organoids on rare and common diseases. So I think it just was a serendipitous match.
It's very cool. Well, let's actually dig into it. I know you touched on what brain organoids are, but let's kind of go into it a little deeper and super digestible for the people who are just learning about it. There's also people listening who probably already have some organoids in their lab. So can you explain what they are and also different ways that you can create them, how long they take, sort of like the process of getting an organoid?
Sure. So if you can kind of imagine when we're looking for drugs to treat a brain disease, the kind of ideal system to test it on would be if we just had a mini version of your brain in a dish, right? Then we could just test all these drugs directly on that model, and we would know if it will work in your brain in kind of the scaled up version. And that's essentially what we're trying to recreate with our brain organoids. And so what we do is, let's say we have a patient with some kind of rare or common brain disease. We can take either a blood sample or a skin sample, and we can isolate cells from those tissue samples. And from there, they undergo this process called reprogramming, which essentially takes those mature cells that you have in your body and it turns them back into a stem cell, which is essentially a cell that is capable of becoming any other cell type in your body. It's called a pluripotent stem cell specifically. And so you could use those stem cells to make any kind of cell afterward. You could make heart cells, you could make liver cells, whatever you want. But in our case, we're interested in the brain. And so we use a specific protocol that we and others have developed, kind of evolved over many years of research to generate brain organoids. And so we treat these stem cells with a variety of different molecules that have been shown to induce stem cells to become neurons and glia and other cell types you'd find in the brain. And there's several ways to make them form these kind of three-dimensional organoid shapes. You can either grow them in a plate that sort of forces the cells to clump up into these little balls. You can also grow them on a little shaker that kind of rotates the plate constantly, so that also helps to induce the formation of that 3D shape. And there's other methods too. It varies a lot between labs and protocols and what's the best for their model system. In terms of timing, it could take a pretty long time to make a brand new brain organoid model from scratch. If you're generating the stem cells yourself, that process can take easily several months and can be quite expensive. Although for more common diseases, oftentimes you can go ahead and skip this step by just purchasing the stem cell lines from a public cell bank or repository. So it's much easier when we have access to those stem cells already from the start. And from there, to make a pretty mature brain organoid that we can use for our studies, it can take anywhere from 4 to 6 months after we have the stem cells to, really get those organoids up to a usable state and also do some basic characterization to understand how they look different from what you'd expect in a healthy brain organoid in this particular disease.
Wow. So you could shake it like a Polaroid picture and it'll grow faster.
Yes, that's just something, it kind of forces them to clump up instead of forming this flat sheet the way you normally would.
Oh, it's very cool. So they are really expensive and they do take a long time. So why is it a smart move, or maybe for an org who's in the beginning, how do they decide where to put those first research dollars? Like, why might investing in organoids give them faster or maybe more relevant insights as opposed to other models that they were more traditionally going toward in the beginning?
Yes, great question. And this was kind of the motivation for us starting Brainstorm and why we think organoids are kind of the future of neuroscience drug discovery, because there's this phenomenon that's existed for a while and has really kind of gotten worse over the last decade, which is called the translational valley of death. And this basically refers to the fact that over 90% of the time, when you take a drug from preclinical work where you've tested it maybe on mouse models or kind of flat cells in a dish, and you try to use that same drug in people in a clinical trial, it fails over 90% of the time. And that's after millions of dollars, sometimes hundreds of millions invested up to that point to get that drug ready. And this is the reason, we've known about, for example, Alzheimer's disease for over 100 years, and there's still no cure. We keep putting these drugs into the clinic and they just don't really work. There's a lot of theories about why exactly this is and why it seems to be especially hard for brain diseases to find a drug that actually works in people. But I think one of the strongest driving factors is the fact that our brains are what makes humans different from every other animal, right? That's why we're the top of the food chain now. It's not because we have bigger claws or can run faster. It's because of our brains. And so I don't think it's necessarily that surprising that the diseases we get in our brains are also going to be very unique to humans. And so when you try to study these diseases in something like a mouse, for example, they're not going to have the same cellular and molecular pathways that humans have, especially when it comes to things like Alzheimer's disease that humans develop naturally, but a mouse will never develop unless you kind of force it to get that disease. And so by using a all-in-human system, like a brain organoid, you have a much better chance of discovering a drug that will actually work when you put it in people. And so if you're going to invest all of this time and effort to generate a model and to discover drugs, you should really kind of stack the odds in your favor by choosing a model where it's all in human, so you don't have to deal with the translation of mouse to people. And ideally, you want a system where the symptoms that you're trying to fix in humans are also present in your model system. So for example, let's say you're studying a type of epilepsy. There are certain kinds of genetic epilepsy where if you take that same gene mutation and put it in a mouse, they may have mild seizures or no seizures at all compared to in people where they may have very severe seizures. In contrast, for many of these diseases, including some of the ones we study, if you take those mutations and put them in a brain organoid, we do see seizure-like activity. We can actually monitor the rate at which the neurons fire in our organoids and see different unique patterns of neuronal firing that correlate with either asynchronous firing or other things that can lead to seizures in people. And so if we can find a drug that reverses that symptom, then you have a much better odds of it actually working in the same way once it goes into a human brain.
It's really interesting. So when you speak of the Valley of Death, where do you see the organoids fitting into that approval process for drugs? And how do you think they can be incorporated into an accelerated approval approach, especially when you guys are also complementing it with AI?
Yes, this is an area that has been evolving very rapidly. And some of your listeners may be aware of the updated FDA guidance that has come out over the past few months and even has been building up for a few years, which essentially states that in contrast to previous guidance, which mandated that you must show your drug is efficacious in an animal model before testing it in humans, now they say that if you can provide sufficient evidence through a combination of organoid or organ-on-chip models and potentially AI as well, you can actually bypass the need for animal studies. And they're actually now moving to say you may also be able to bypass some amount of the toxicity and safety studies, again, where you can show sufficient evidence in your in vitro or cellular system that your drug is safe. And so I think this is going to be really amazing progress for brain diseases, especially for diseases that we just know aren't modeled particularly well in a mouse system or in other animal models. And it also allows us to accelerate this pathway from your initial drug discovery into approval to go into the clinic. So in our case, we can grow our organoids in pretty high throughput. We have these plates with 384 little wells in them, and we can put one organoid per well. So that means we can screen 384 different treatments at a time, just in one plate. And we can do, multiple plates in a day. And so this allows us to test drugs on this system that's pretty much as close to human brain as you could get in that kind of scale. And then based on this FDA guidance, as long as you can show it's efficacious and you can prove that it's safe, you can go directly into the clinic without having to take the trouble and spending often years developing a mouse model for this disease and testing its efficacy. And especially for rare diseases where there probably isn't a mouse model already available, this can greatly accelerate how much time you need to actually get a drug into the hands of patients that are desperately waiting for it.
So I've always heard that, brain organoids would complement and not replace things like mouse models, but you're saying that it potentially genuinely could.
Yes. And we've actually seen the first early examples of this occurring at the precursor to our company, Effie and Maya, which used the same platform as we do it here at Brainstorm. They actually were able to discover a repurposing drug for Rett syndrome. It was actually the drug Aricept that's commonly prescribed for Alzheimer's disease. They found that in Rett syndrome organoids, this actually fully rescued the seizure-like activity that they were able to see. Rett syndrome is another kind of rare genetic epilepsy that we've been studying. And they went from doing this initial repurposing screen into being ready for a phase 2 clinical trial and filing the clinical trial application in just 9 months, which is dramatically faster than you would typically see. And they didn't require any additional animal data to get to that point. We actually just heard about a month or two ago now that we've received clearance for the IND, which essentially means the FDA has given us approval to take that into a phase 2 trial. Now, again, that's a repurposing drug, right? So that, it already has some pre-existing data, But I think it is a really exciting first step to show that as long as you can make a strong case for your drug using organoid data alone, the FDA is willing to consider moving it forward into clinical trials without having to go through mouse studies.
Wow. Okay, so I don't know if you know Tommy Pham, his kid named Raiden, but I was reading something that he just released. His kid has UBA5, and they were talking about in the article that organoids can test different areas of the brain, but how the brain can respond differently even if you have organoids of kids with two of the same exact mutation. So that makes it even like kind of blow my mind a little more that you could actually really tailor them, like tailor treatments to these kids who you think that this one drug will work for both of them, but they might not because their brain is responding differently. Can you sort of just explain that, making my brain explode? Like, that seems like it's actually harder and way more complex, and now maybe nothing is really going to be the answer for everyone unless we test every single one separately. But yeah, I guess if you already have them, maybe it's better.
Yeah, exactly. I think this is one of the areas that we have strong potential for brain organoids, because there's been this evolution in thought where, people say Alzheimer's disease or Parkinson's disease. But nowadays, we don't really think of these kind of conditions as diseases, but more like syndromes, where there's a very complex mix of genetic factors, environmental factors, lifestyle factors that all kind of make this unique combination that leads to a specific person's development of that disease. And so it's unlikely that any one drug is going to be effective for every single patient, even if they ostensibly have the same disease. And so we need a better way to model each person's brain and understand the different variables that contribute to their brain and why it's having this particular disease. And this is where the growing field of precision medicine comes in, which is trying to make drugs that work for a particular subset of patients, not necessarily for every patient. This is an area we've been working on quite a bit. In particular, we just launched a new collaboration with the Cure5 Foundation, which is a nonprofit dedicated to a disease called CDKL5 deficiency disorder. It's another kind of rare neurodevelopmental disorder that leads to seizures and other kinds of cognitive impairment. And we're actually working with two specific patients, stem cell lines, so just two girls who have CDKL5 deficiency. And we're developing brain organoids from these two lines, and we're going to test repurposing drugs the same way that was previously done for Rett syndrome, but now for CDD. And this is going to give us a lot of insight into, will the same drugs work for different patients with the same disease, or are we going to need to take really a precision medicine approach and pursue different drugs through different people who have this disease? And I think this is the huge potential of brain organoids, because modeling every single different mutation in a mouse model is just not practical. You're never going to get that many mice with hundreds or thousands of different diseases. But once you have a pipeline set up to generate brain organoids, you can really bump up the throughput and make a precision medicine approach possible. So I can imagine a future where if you have some kind of brain disease, instead of just going to get the same drug that everyone tries and maybe having to go through many different iterations of treatments before you find one that works for you, there can actually be some form of a test, maybe a genetic test, maybe something else, where that will determine what subtype of the disease you have. And they can either give you a drug, if it already exists, that's tailored toward you, or maybe make a new model of your brain and figure out what drugs are going to work best in that model system.
Wow, that's very exciting, and I hope to closely follow the CDKL5 stuff. And yeah, that makes me just think about sort of the ripple effect, right? And like how many other disorders it can sort of affect. And you said something about it being, did you say platform? Like making organoid research sort of a platform?
Mm-hmm.
Can you explain that a little bit?
Yeah, I think organoids have really evolved in the past 10 years. They were really starting out as this really academic focused topic where the goal was to just model human brain as closely as possible, no matter what. And the problem with those kind of systems is they were not going to be amenable to a platform. In other words, they were not reproducible enough to actually screen drugs in a high throughput way. These models are highly variable every time you start to grow organoids. And this makes sense, right? The human brain development in utero is also somewhat random. And so for academic labs, that's totally fine to have this random variation. But if I'm trying to grow thousands of organoids, I want them all to be identical so that I can tell which drugs are going to be effective and which aren't. And so That's part of what secret sauce at our company really is a method to grow these organoids in a way where they're going to be extremely consistent and extremely robust. And they're going to all have the same symptoms that we want to measure in our assays. And that essentially allows us to build this platform that lets us screen drugs in a very high throughput way. And also, now that we have these protocols, it's much faster to create and characterize new disease models because we already have that experience under our belt.
I'm really, really excited, and I kind of want to take a tour of your lab when I'm down there next time.
Of course, you're always welcome.
So obviously things evolve and they're always getting better. What are some of the technical limitations now that you're dealing with? Like, what can organoids not tell us? And what do you think is getting implemented to maybe make that better?
Yes, every system has its limitations, and I definitely don't want to claim that organoids are a silver bullet that will solve all problems. One of the limitations of our model currently is that we don't currently have a cell type called microglia, which are essentially the immune cells of your brain. And the reason we don't have them is because microglia actually come from a completely different cell lineage compared to all of the neurons and glia in your brain. And so it's possible to add them in the process. You'd have to essentially grow them separately and then sort of mix them in to your organoid during the growth period. And that's something we're actively looking to do, especially for diseases where the immune system appears to play a larger role. So I can kind of see that being a newer generation of organoids that we'd be very interested in looking into. Another area that we currently can't model, at least with our system, is the blood-brain barrier, which is exactly what it sounds like. It's a barrier that prevents drugs and other kinds of toxins and infectious agents, anything from passing from the blood into your brain. And this is a really important system. It's really good to prevent stuff you don't want getting in the brain, but it also can prevent drugs that you do want to get into your brain from passing. And so our organoids don't have this system currently. And I think this is actually an area where organ-on-chip technology could be a complementary technology. Organ-on-chip is similar concept to organoids, but they're usually lower throughput. And they're meant to model these more kind of complex, multi-layered systems like the blood-brain barrier. So there's multiple labs and companies creating these models of the blood-brain barrier to test how well drugs can penetrate. And there's other similar organ-on-chip models for looking at liver toxicity, looking at distribution, looking at pharmacodynamics of your drug. And so I think organoids, to me, are a really promising system to look for how efficacious your drug is, how well it actually fixes the thing you're trying to fix in a given disease. But we're still going to definitely need other specialized complementary systems before we can make sure that the drug is going to actually perform as we want before we put it into a human.
It's always the blood-brain barrier. It's always that.
It is tricky.
Barrier. So what's Brainstorm working on right now that you're super excited about?
I think one of the areas that I'm really excited about and I think is really promising is the work I briefly mentioned earlier on bringing AI into the picture. I think every company is interested in implementing AI at this point. It's kind of causing a revolution in the tech and biotech spaces. But a lot of the innovation I've seen with AI lately has been focused very much on biochemistry. It's basically trying to find a molecule that will bind to some target that you think is important to your disease. And these tools work pretty well, actually. But the problem is that the targets we're looking at actually tend to be wrong in a lot of cases. Think of, again, Alzheimer's, where we've been trying to get rid of this toxic protein amyloid beta for 100 years. And it turns out that actually doesn't work. You can completely get rid of amyloid beta, and that doesn't actually fix the disease. Similarly, for other brain diseases, we can make a drug that binds to your target, but how do you know your target is actually correct? And by target, I mean a protein, typically in your body, that has something wrong with it. And so in our case, instead of just using AI for this chemistry approach, we're really taking a biology-first approach to AI. And so we're using this technology called foundation models, which is a pretty new type of AI framework, where essentially you can take these gigantic single-cell RNA sequencing data sets, so taking millions and millions of cells and sequencing their RNA, and you can build these really large AI models to understand the relationships between all of those genes. And what we can do at Brainstorm is take these large models that other people have published, and we can fine-tune the models with our own brain organoid data. So essentially taking this, extremely smart model and then telling it solve this very specific problem using this very specific data that we're giving it. And we can use these models to do all kinds of things. We can use it to identify targets that are more likely to be involved in a disease without having our own human biases play a role. We can also use this to stratify patients for clinical trials and predict which set of people our drugs are most likely to be effective in. You can even do things kind of like a virtual cell approach where you could simulate, for example, a gene knockout before you actually try it in your cells. So you could basically delete a gene from your system and say, okay, how does the cell respond in this virtual environment when I get rid of this gene? And that can give you insights on its function as well. So this is a really exciting area that we've been pursuing. We actually won an award from NVIDIA, the computational company, last year for this work, and we're collaborating with NVIDIA now to improve their implementation of our pipeline. So I think this is going to be kind of a new generation of biotech by implementing AI in this way, and I'm really excited to see the progress we're able to make in the future.
This is really exciting. Like, I kind of want to just sit down and listen to this stuff for a while because it's really kind of mind-blowing. And as, you know, a rare disease parent who is on a board who's constantly trying to figure out how we can move faster and get better insights and make better decisions, it just really makes me think a lot. So are you saying that, like, Brainstorm Therapeutics will travel? Like, so a family decides to invest in brain organoids with Brainstorm Therapeutics. Do you travel through the entire process with them? So you're doing the brain organoids, you're testing the drugs, then are you applying to get clinical trials going? And then, like, are you following that whole process, or is at some point there like a handoff of the service that you complete and then they go and do the next step somewhere?
Yeah, it's evolving over time. And it also depends on whether we're looking at a repurposing drug or a completely new drug. In general, we definitely will take the project, for example, again, going back to our Cure5 project, we're starting again from scratch, from brand new patient stem cells that have never been characterized. We're growing organoids. We're going to figure out what exactly kind of seizure-like activity they may be having. We're going to screen several hundred repurposing drugs for that project. And then we're going to also implement the data we get from these models into our AI work to figure out what other targets might be useful if we choose to pursue a new drug instead of just a repurposing medicine. From there, it kind of depends on what the foundation would like to do. In general, it's very hard for a small company to completely manage their own clinical trials. These are an extremely expensive process and also take a lot of expertise in that area that, you know, is hard to recruit if you're, you know, small team of 3, as we are right now. And so a lot of companies what will they'll do at that stage, and what we're planning to do most likely is if we uncover a new therapeutic candidate of some variety and we've characterized it, maybe we even have approval from the FDA to start clinical trials, you can essentially license that drug to a much larger pharma company who can handle the clinical trial work. And so that makes sure that the drug is actually getting developed and not just sitting on a shelf somewhere, and it frees up resources back at Brainstorm to move on to the next discovery for a new disease you want to study.
Yeah. Wow. Cool. So, and we don't have to necessarily record this part, but I do wonder. So if a rare disease organization comes to you, say they have 400 patients and they want to invest in some brain organoids, they get a hit, it moves on and it moves on. Is there like shared IP with these orgs, like from the beginning, or like, how does that look on paper with the money that's being spent and the data? Like, is stuff shared back with the orgs fully? Are you a partnership?
Yes, and this is something that we negotiate individually with each group that we work with. So we're certainly open to discussing with every group kind of what their priorities are in terms of sharing an IP. I will say that in terms of data sharing at the minimum, we're completely transparent with that. Any kind of new data we generate is always going to be shared with the partners we work with. In terms of IP, we're still in the early stages of figuring this out on our end. There aren't a lot of existing models about there in terms of IP sharing and potential revenue sharing between nonprofits and businesses. But this is an area that we're extremely interested in. And we think it's also an area that many nonprofits are interested in exploring. So I would say we're open to anything. And if any groups are interested, they should definitely ask us. And we're happy to discuss on an individual basis.
Awesome. Thanks for that answer. So if a small rare disease organization does want to fund or initiate some brain organoid work, what are some of the first steps that they can take?
Yes. So the first step is to make any kind of brain organoid model is always going to be developing stem cells. And so if you already have any stem cells available for your disease, that already cuts several months and a lot of the cost off of the timeline. If you don't have your own stem cells, we can also make those. And so the first step in getting ready for that would be to find patients who are willing to give either a skin sample or a blood sample and have us create stem cells from there. After that, as I said, it could take anywhere from 4 to 6 months to completely characterize these organoids we developed, and it can take a couple of months after that to do a small drug screen. So it really depends on the scale of the experiments that an organization would like us to do, and also the depth of characterization that they'd like us to do on their organoids.
Okay. Is there like a flat fee of any sort, or is it different depending on everyone's budget and how many samples and everything that you need?
It varies a lot. Every group we work with has different numbers of patient lines they want to do, different types of drug screening, different experiments to characterize the organoids. It's generally in the ballparks of like the 6-figure range, but within that range, it can vary a ton. For an experiment where you just have maybe one patient cell line and you just want to grow organoids and kind of see what they look like, that's going to be a much lower cost experiment. Whereas if you have maybe 10 or 20 different patient cell lines and you want to screen thousands of drugs, the costs are orders of magnitude higher. So it's something we really talk about in depth with every individual group we work with. And another thing we try to prioritize is setting up milestones for these larger projects. Especially for a smaller nonprofit, they often can't front the entire cost of a very large, sometimes multi-year project like this the way a much larger group could. And so instead, we set up these interim milestones where we say, okay, maybe after we've fully characterized the organoids, we'll have a discussion to say, do these phenotypes look good for us to go ahead and do a drug screen? If so, there'll be a new milestone payment. If not, we end the project and they don't have to pay anything further. There's always some level of unpredictability when it comes to making a brand new organoid model. And we don't want these nonprofits to have to commit to a much larger project if we don't even know from the outset if we're going to see the exact symptoms that we want to see in these organoids. So we'd like to keep things pretty flexible and customize our contracts depending on what each individual group is looking for.
Yeah, I love that. It's always moving. And this might be a dumb question, but you also have to pay for like storage to keep these organoids alive to continue to do tests on them? Or is that something that's kind of worked into it? Like, is there like a shelf life for how long these little babies can be alive?
Yeah, so a lot of times what we do is organoids, you can't keep them in culture too long, just mostly because the symptoms that they exhibit can change over time, and you want to keep things pretty stable if you're going to do experiments on them. And so a lot of times what we do is we'll just store the stem cells that can be kept in a freezer forever, essentially. And you can stagger your organoid preps over time. And so, for example, let's say I need some organoids to be ready in 2 months for a big experiment. I might start some today, and then I'll also start another batch in 2 weeks so that I don't have to start completely over again if I want to do another experiment. So we don't really store the organoids. They're a little bit hard to preserve and thaw the way you could with a typical stem cell, but we do spread out the organoid prep so that we're not wasting too much time just growing them from scratch.
Cool. Okay, that makes sense. So I know you're obviously deep in the science, but I wonder if you have ever had the opportunity to like sort of work closely with families, or if you have sort of like a personal story that sort of stays with you and like helps you in the lab.
Yes, I've really been enjoying getting to meet all of the leaders of the Cure5 Foundation. So Andreas and Anna and Lily, we've met all of them multiple times in person and on Zoom and talked about their experience, having children with this very rare and very challenging disease. And I'm always extremely impressed with the steps that these parents have gone to for these kids. They will always be way more knowledgeable about their disease than me. I have a PhD in neuroscience, and yet they could educate me all day about this disease. This is their life. This is something they study way more than I could. I have to learn about every disease a little bit, and they have to know this one disease in extreme depth. And they've also just really been inspiring to see how well they work with other rare patient organizations. That's something I've really been impressed by with the rare disease community, is that people aren't siloed to just their one individual rare disease. They've really come together and learned from each other and shared strategies with other organizations about how to raise funding, what's the best way to do repurposing screens versus new screens. And I think that's kind of refreshing, especially compared to, sometimes in larger pharma or when you're studying a very common disease, there can be a bigger sense of competition in those spaces. And I think in the rare disease space in particular, it's really more about collaboration and learning from each other. So I've really enjoyed getting to meet all these people and to feel like the work I'm doing can actually have a very direct improvement on someone's life.
Awesome. Good job.
Good job, Maya. And yeah, Andreas and Anna and Sienna, they're a beautiful family. And I'm so happy that they have this opportunity with you guys. I think it's going to help a lot of people, and definitely just move things forward in terms of, other opportunities that rare orgs can take, right? Like sometimes it's just that nobody's doing it, or only some can invest in it, and it's too scary, or it's too expensive, or people are too skeptical. So I really, really just hope for the best for this situation, because I love seeing new doors open for all these rare disease organizations on different avenues they can take by being inspired by another family, right, who took the plunge. So that's really cool. Maya, is there anything I didn't ask about brain organoids that I should have for this episode?
Let's see. I think you asked most of the important questions. I guess I'll just say that for brain organoids in particular, but also just for all sorts of companies looking at rare and common diseases, it's a very challenging environment to be doing drug discovery right now, again, especially for rare diseases. I'm sure everyone's aware of the cuts to federal funding and also the challenges with raising venture capital right now. And so I think this is a time when nonprofits and other patient foundations can really play a strong role in shaping which disease areas these new companies focus on. This is a space where historically we've relied a lot on federal funding, especially for earlier stage research and also for research where it's, sometimes the diseases are so rare that it's hard to create any commercially viable drug. And that's where grants have stepped in to fill that role. It remains to be seen whether we will be able to continue rare disease in general at the same rate without federal funding. And so I think now, if your organization has funding and has been kind of waiting for the right time to deploy it, I think now is a time when a lot of biotech companies are looking for alternatives to federal funding. And so especially if you have a rare disease that it's challenging to convince people to work on, sometimes just providing a grant to get a company to start looking at it and to see how well it performs in their model, that can play a major role in determining where they actually take their pipeline in the long term.
Well, thanks for being my guest, Maya. I'm really sort of inspired to just kind of go nerd out a little on this, and it's fun to be excited. It's fun to be excited in this world. So thanks for all of that information. Thanks for what you do at
Yeah,
Brainstorm.
Thanks so much for having me.
Yeah, and congratulations on that award, by the way.
Thank you.
Yeah, I'll have everything listed in the show notes. So you'll be able to contact Maya and them if you have any other questions. But very cool. Thanks for the crash course, and thank you for being my guest.
Wonderful.
I hope you've been enjoying this podcast. If you like what you hear, please share this show with your people, and please make sure to rate and review it on iTunes or wherever you get your podcasts. You can also head over to Instagram, Facebook, and Twitter to connect with me and stay updated on the show. If you're interested in sharing your story, or if you have anything you would like to contribute, please submit it to my website at effieparks.com. Thank you so much for listening to the show and for supporting me along the way. I appreciate y'all so much. I don't know what kind of day you're having, but if you need a little pick-me-up, Ford's got you.